All right, we're staying in Texas. Moez Dawood from Baylor is going to talk about some saturated genome editing work.
Thank you. So, Moez Dawood, hello again. Baylor College of Medicine, Houston, Texas, co-mentored by Dr. Richard Gibbs and Jim Lupsky, but currently working in the lab of Dr. Leah Starita at the Brodmann-Beatty Institute of the University of Washington via the Center for Multiplex Assessment of Phenotypes internship program. So one of the main foci of Dr. Starita's lab is to use high-throughput functional genomics to study VUS in hereditary breast and ovarian cancer predisposition genes. So along these lines, the main gene that we're doing right now is PALB2. So PALB2 functions in homology-directed repair of double-stranded DNA breaks and promotes nuclear localization of stability of BRCA2. Pathogenic variants in PALB2 are implicated in breast, ovarian, pancreatic cancer, as well as a biallelic rare disease trait, Fanconi anemia. Last year, PALB2 was elevated to the ACMG73, meaning that secondary findings in PALB2 should be reported to patient providers as it may have clinical implications. Further, in ClinVar right now, 99% of PALB2 missense SNVs are VUS, emphasizing a need to do functional characterization in this gene. We performed saturation genome editing of PALB2 to investigate the effect of all possible 10,683 SNVs in PALB2 on cell survival. SGE allows for CRISPR-Cas9 mediated high-throughput functional assessment of every SNV's effect in the endogenous loci of an essential gene based on a Gestalt readout on cellular viability. Compared to similar methods, SGE will not only inform about all missense, nonsense, and synonymous SNVs, but also canonical and non-canonical splicing SNVs and select intronic SNVs. Here, in data from PALB2 exon 2, you can see a bimodal distribution of the functional scores of each SNV. On the x-axis, you can see the actual functional score for each SNV. On the y-axis, you see a count of the number of SNVs within a bin of functional scoring. The lower or more negative the functional score, the more on the left, or the more deleterious the variant. The two sides of the bimodal distribution are defined on the left as known deleterious SNVs, that is the nonsense and canonical splicing SNVs, versus on the right, synonymous SNVs. Additionally, we show how the bimodal distribution changes by prolonging the experiment from day 11 to 14 to 17. Due to the way we've advanced the technology and the prolonged time course, we're able to delineate a subpopulation of missense SNVs from this tail that comes out over the course of the experiment. And so this subpopulation of missense SNVs is breaking away from the right side of the distribution, or the side with the variants that are not killing the cells in the assay, and migrating towards the left side, or the side of the distribution that are killing the cells in the assay. And so much like actually Dr. Kitzman's talk this morning and probably what Dr. Hurls will talk about later, we hypothesize that SGE is a sensitive enough assay to detect putative hypomorphic missense alleles. And we do not think that this is noise in our functional data because we can see these functional scores replicate across independent biological replicates with pretty good R-squared. So within the N and C terminal functional domains required for binding RAD51, BRCA1, BRCA2, we're seeing over 10% of missense SNV alleles result in decreased PALB2 function that compromises cellular viability. These SGE-derived functional scores are currently 100% consistent with published orthogonal functional assay data from the Van Atticum group, and we will hear updates from them tomorrow as well. Finally, we can reproduce known genomic phenomena such as escape from mRNA surveillance mechanisms. For example, we show here that stop gain as well as stop loss variants in the last exon of PALB2, which normally accumulate on the left side of our binomial distribution, are actually spread out across the distribution, demonstrating that nonsense SNV alleles can escape from nonsense-mediated decay, and stop loss SNV alleles can escape from non-stop decay, as predicted for the last exon of multi-exon genes. So in total, we are finishing up SGE of PALB2. We have a couple exons left. Thus far, we're seeing about 10% of missense SNV alleles are functionally abnormal, And we're hoping our data can be very useful for PALB2 variant interpretation moving forward. Thank you.
Go ahead.
Thank you. Great talk. I just wonder, how did you determine the size of your target region, and also how did you normalize your replicate experiments?
Yeah, so there's a min-max normalization that happens within each exon and then across the entire data set when we accumulate all the data. The functional score itself is basically just a ratio of the number of sequencing read counts seen at a later time point in the experiment to what's seen early on in the experiment, or in this case, what's in the library itself.
So did you just average your replicates?
Yeah, there's a level of averaging, but there's also a lowest regression to correct for positional biasing, and there's other statistics implemented to take out other biases. Yeah.
Thank you.
Have you had a chance to look at actual clinical outcomes of people who have PALB2 variants?
Not yet, but we have been collecting phenotypic data to eventually do that. We've also been collecting genotypic data to eventually reinterpret, hopefully, some VUS.
Great.
Thank you.
Perfect. Thank you.